Hello, my name is Hugh Fan. I am a professor at the University of Florida. My contact information is listed on this title slide. Today, I will briefly discuss microfluidics, which is the focus of my research, as well as those activities related to the MIST Center. The MIST stands for Multifunctional Integrated System Technology. Several current and past MIST industrial members have expressed an interest in microfluidics. This is outline of my talk. I will briefly introduce microfluidics, a field started in 1990s and exploded in the last couple of decades. I will then present the research project examples in each aspect of microfluidics, including microfabrication, microflows, and applications. Finally, I will close my talk by discussing our efforts in flexible, stretchable electronics that are a part of the MIST Center. I will talk about device fabrication using a tattoo making tool and about integration of electronic components on flexible substrate. Microfluidics is to study fluids in the micro scale. The field involves three aspects. The first is microfabrication, which is to make devices containing microscale features. The related activities are listed here, including device design, photolithography, molding, lamination, 3D printing, and manufacturing. The second part of microfluidics is microflow, which is to study the fluids in the microscale. The activities include studying the fluid's behavior in small scale, such as testing and simulation, flow controls, such as pumping and valving, and the platform integration, such as macro to micro interfaces. The third aspect of microfluidics is applications. Potential applications of the portable miniaturized devices include point-of-care testing, such as at home or in emergency rooms, environment monitoring, and the detection of pathogens or biofare agents in the field. Microfluidics is promising to reach holy grail of the lab on chip, i.e. shrinking a lab into the size of microchip, computer chips. In analogy to shrinking a computer from the size of a room in the 1950s to a laptop today, instruments for chemical and biological analysis could be miniaturized using modern microfabrication technologies. Another term you may hear is microTAS, standing for Miniaturized Total Analysis Systems. In terms of microfabrication, silicon is a substrate of the choice for semiconductors, and it has also been used for fabricating microfluidic devices via photolithography. Photolithography was later extended to glass device fabrication. Thermoplastic manufacturing process, such as molding, have also been used for making plastic microfluidic devices. Other substrates for making microfluidic devices include elastomers and paper. As an example, we have worked on cycloolefin copolymers, COC, as a substrate to fabricate thermoplastic microfluidic devices. The process is similar to manufacturing compact disc or CD. On back of the CD, there are microgrooves, which have similar dimension to that of microchannels in a microfluidic devices. A piece of CD costs less than $1, and the manufacturing cost is about a few cents. The low cost is critical for some applications, such as medical device diagnostics, which requires devices to be disposable to avoid possible cross-contaminations between the samples. The figure is a picture of the device made from COC using compression moldings, and the device was designed to perform two-dimensional protein separation. The interface between two dimensions is shown on this the explorer view on the top right, which was obtained by scanning electron microscope. The complete device filled with a fluorescence solution shows a two-dimensional channel interfaces. The fluids in microscale can behave differently from those in the macro scale due to their low Reynolds numbers and high surface to volume ratios in microchannels. The activities we have worked on is in this aspect of microfluidics include pumping, mixing, simulations, and microvalve development. One example shown here is to design a device for tumor cell isolation from peripheral blood of cancer patients. The device contains an array of the retinal filters. Shown in this is a journal inside a cover page cartoon. A sample into the inlet of this device interact with those retinal filters while the main flow will go through this serpentine channel. The simulation really help device design, which we call a parking lot-like arrangement. When a filter is occupied a tumor cell, the subsequent cell and then flow will change that directions to interact with next filters illustrated here. This device design prevents cells from clogging all the channels or the filters observed in the conventional filter devices in which the flow of filtration are in the same direction. One of the microfluidic applications we have worked on is Zika virus detection at the point of care. In this cartoon, the sample will be processed in this 3D printed sample preparation device, which is pre-packed with the lysis buffer, binding buffer, and the two washing buffers. Virus lysis, RNA enrichment, and washing are carried out sequentially by sliding the bottom unit around this top buffer unit. So valve activation mechanism by sliding is shown in this side view diagram. It is similar to a ballpoint pen in which ink is dispensed onto paper when the ball at the tip of the pen is pressed while writing. The ball at this bottom of the reservoir essentially prevents the fluids from flowing out. Then when you're sliding the bottom one, body unit, the pin in the bottom unit will push the ball up and the fluids flows down. This experiment can show on these figures that red fluid dye here, and then when bottom sliding to the left, the solution will move flow down to that bottom unit. The enriched virus, the virus DNA here will go through this so-called isothermal nucleic acid amplification method so this is called amplification. The resultant products are detected by colorimetric method, a color change observed by naked eye or recorded by a smartphone camera. A blue LED here, the blue LED flashlight can be used to enhance sensitivity. The smartphone photos of either ambient light or blue LED light shown here. So device 1 and 2 contains that either 1 PFU or 0.1 PFU. PFU stands for the plaque forming units of those Zika spiked in human urine samples, while device 3 is a negative culture. You clearly see the color difference between the negative and the positives. We have used a similar device to study correction and detection of airborne, the viable airborne viruses, basically influenza viruses. Recently, we studied the SARS-CoV-2 related to the current COVID-19 pandemic, with a focus on that simultaneous detection of both SARS-CoV-2 and influenza viruses. We also developed a related device for E. coli detection for monitoring water quality with the potential to look at that Chlorella brevis, also known as red tide, which is a problem around the Florida coastline. Now I'm shifting to our effort on flexible stretchable electronics. To make tattoos, we need inks or dyes to print them onto paper or body. To create flexible stretchable electronics, we need to print contact ink or the like onto substrate. This is the reason we basically have been wondering if we can using tattoo making method to fabricate flexible electronics. This figure shows a picture of that the airbrush, which is a device often used in tattoo shop for creating the body arts. This figure here based on the principle and Venturi effect. When compressed air gets into here to create a vacuum near the nozzle, and the vacuum will suck the reagent from the reagent cap tube to the nozzle and then spread out. And then air is controlled by this trigger, and the inside of a needle, which is controlled by the control screw, which dictate how much the opening here, and then basically how much reagent can be spread out. We used airbrushing to create various devices as the patterns on paper, thermoplastic thin film, and PDMS or polydimethylsiloxane. This figure shows an array of reagent dots, and then here is a trace of the two letters U and F using a protein solution as the ink. We also used airbrush to fabricate microelectrodes and then use them for electrochemical detection. Similarly, we used photolithography to fabricate these micro heaters on the micro thermoplastic thin films, and that also can be integrated in the microfluidic devices. We also printed the capacitors of that interdigital shape, okay, interdigital patterns on the two frequently used flex substrate, TPU or PET or thermal plastic polyurethane or polyethylene terephthalate. As expected, we observed a linear relationship between the capacitance of the device and the number of that fingers. Towards the goal of flexible electronics, we demonstrate a device with printed conductive trays and then capacitive touch sensors, followed by attaching battery, LED lights, microcontrol unit, switch, capacitor, and resistor on that flexible, stretchable TPU. And then after we press this, the sensors, using fingers, that corresponding LED is lighted up, indicating that functional device. We also fabricated and characterized many other devices. For example, a multi-layer flexible printed coil is shown on this, which can be used to generate low-frequency magnetic fields for applications such as electrodynamic wireless power transmission. Compared to traditional coil forming methods, screen printing enables rapid fabrication of thin, lightweight, conformable, and flexible structures. The flexibility is demonstrated by repeated bending of the device shown at here, bottom, up to 10,000 bending cycles. We also demonstrate wireless power transmission of 1 milliwatts of power at a distance of the 4 centimeters between the flexible receiver and the transmitter. To conclude my presentation, I have 3 take-home messages. 1. Microfluidics is a very promising and diverse field. 2. There are many interesting and impactful research topics in each aspect of microfluidics, including microfabrication, microflows, and application. 3. Flexible and stretchable electronics can be found in industry and medical devices in your everyday life, with a huge potential in health monitoring for aging populations and in reducing healthcare costs through preventive care. I would like to thank all my former and current students and postdocs for their efforts and dedication, and particularly these students who made contribution to the work I have presented here. I'm very grateful to my collaborators and their labs for their support and teamwork. I acknowledge the funding from these agencies, foundations, and institutions. If you have any question or need more information, please check my website or send me an email. Thank you for watching.